many thanks for the organisers to invite me here today. And I'm going to be talking about some of the work I've been doing on blood substitutes and called here Pint of Artificial Blood. We've not quite produced that amount so far, but we're getting there. But first of all, I have to ask everybody, anybody got haemophobia? Fear of blood? I have had a few people faint. Okay. So I won't be talking about blood and blood products, but I won't be showing any real blood. But Hemo2 is a biotechnology project at Essex, and we've been going at it for a few years now, trying to generate a new generation of blood substitutes and oxygen therapeutics. I'll tell you a bit more about oxygen therapy. I mean, probably everybody knows what a blood substitute would be, but maybe not everybody will know what an oxygen therapeutics was, but I'll come to that later on in the talk. But the thing is, it would be great if we had our own blood bank, own personal blood banks, but we can't really have that. A few people, very important people, do have that apparently. And apparently the President of the United States has their own blood bank they carry with them when they go abroad. And this was from a few years ago. This is from, this is from the Daily Mail, so I'm not exactly sure how accurate this is. But if you notice, I mean, this was when President Donald Trump visited the UK a few years ago. And the beast in the car, among things like the gas grenade launchers and the pump-action shotguns in the doors, reported that had blood bags of the President's RH negative blood. So it's carried in the car with them just in case. And it's also reported, this is the Sun, so again. So no, I don't read these papers. But in the Sun, they report that when the Queen went on royal tours, they had their own blood with her. I'm not sure if it's her own blood, but probably some blood which was compatible. But the rest of us, we can't have our own blood banks carrying around with us, and we have to rely on getting blood from hospital if we need transfusion. So why is there a need for artificial blood? Well, a lot of countries, the population is getting older, so we need more blood donations. We need more transfusions being done. There's about 40 million transfusions in the world per year from about 110 million donations. And demand is going up and up and up, but the number of people who are able to give blood donations is going down. And this is partly because of less young people being around in a lot of countries, but also because of the modern health issues like diabetes and high blood pressure. So if current trends continue, there will be an issue in the future with the supply of blood in many countries. Now, blood transfusions is very safe in this country, but in other countries such as Africa, where there's a prevalence of HIV in the population of about 4.1%, and some areas above 10%, getting blood for blood transfusions is a lot more difficult. And there are those that refuse to have blood transfusions for cultural, religious reasons, or may be immunocompromised. So they may have an immunologic disease, or they may be on chemotherapy, radiotherapy, reduced ability for immunogenic compounds, and they can't have those donated bloods. And there's issues with blood storage as well, and I'll talk about that in a minute, but after you transfuse blood, it doesn't really work the same way that normal blood does for about 24 hours. It needs time to get settled back in to the routine of delivering oxygen around the body. So, and there's also a tainted view, especially in this country, of blood donations, and this stems from the 1970s and 1980s, but still got ramifications today for these tainted blood scandals for hepatitis C, HIV, and a few others. And in the 90s and 2000s, we also had issues and worries about prion disease being transmitted through blood donations as well. And we don't know what the next blood-borne disease will be. Fortunately, coronavirus is not known to be transmitted via blood transfusions, but it could be something around the corner that we don't know about. There's always a window between the sort of detection of these diseases in the population and tests available to be able to show if the blood is safe or not. Okay, these windows are getting shorter and shorter, but there's still windows for when a new blood-borne disease occurs for when it can be tested. And as I said, stored blood is not normal blood. If we store blood, and I'm talking about red blood cells here, over the course of a few days to a few weeks, then the biochemistry of the red blood cells change. And things like lactic acid builds up, so pH goes down, so it becomes more acidic. And this molecule called 2,3-diphosphoglycerate, which is needed to control the ability of haemoglobin inside the red blood cells, which carry the oxygen around the body, that attenuates the ability of haemoglobin to be able to carry oxygen or not. And it takes about 24 hours for all this biochemistry to re-establish itself once these red blood cells are transfused into a patient. And there's also morphological changes in the shape of the red blood cells. So you can see here some typical red blood cells, and they're very identifiable, these bicon-cave shape. And after about 35 days, you can see there's a lot of these red blood cells that change their shape to these spiky cells. They're called echinocytes or Burr cells. And after 42 days, most of them are these echinocytes. And these are very rigid cells. One of the unique things about red blood cells is that they can press. They can get into capillaries, which are only a third of the diameter of a standard red blood cell. But these echinocytes, they can't do that. They have to be broken down by the liver and the spleen because they don't work properly. And the FDA approves blood transfusions up to 42 days, but in the UK, we've taken that down in the last few years from 42 days to 35 days. But there's still issues in the change of the morphology, the change in the biochemistry of these red blood cells. So at Essex, we've had some funding, mainly from Medical Research Council, but also from BBLC and a few others. I'll give acknowledgements at the end, of trying to use genetic engineering to produce an artificial blood substitute. And there's lots of different avenues of research for artificial blood substitutes. I don't have time to go through them all, but the one we're focused on is haemoglobin-based oxygen carriers, HBOCs. So these HBOCs are made from haemoglobin taken from the red blood cells. And if we could use that without having to use the red blood cells, it would be very advantageous because we could keep that at room temperature. If it's sterile, we do it sterile, so we can keep it at room temperature. We can keep it, instead of having it refrigerated, we can keep it up to, well, certainly for several years, whereas 42 days, 35 days in the UK is the maximum we can store red blood cells for transfusions. It's ready to use. We don't need to do any testing. Okay, some of you might say, well, can't we just use O-negative blood, which is the universal blood type? Then, well, only about 10% of the population are O-negative, and throughout the world, that's much less than that. In some countries, it's only a few percent O-negative. And lots of people do have atypical antibodies, so they still have to be tested, even if they're given O-negative blood. If we generated it through our biotechnology through fermented bacteria, and I'll show you a bit more about that in a little while, we could produce vats and vats of it in huge fermenting containers, Whereas now, of course, we have to use human blood from donations for the blood transfusions. And if we use these fermented bacteria, we can guarantee the purity of the haemoglobin, and there's no diseases, no viruses that these bacteria pick up. Whereas for the NHS, they selectively test for various diseases, but they don't do a huge amount of testing. That's why you have to do quite a long, if you go for blood donations, you have to do a long list of things that you've not done, have done in the recent time to minimise the chances of getting diseases. If you've been to the Amazon rainforest, for example, then it could potentially get some diseases back from there or some other areas. And of course, with blood substitutes, we don't have to worry about things like immunocompromised patients, and we don't have to worry about religious groups, because we did a study as part of a EU Framework 6 project quite a few years ago now, and they discussed the issues with people like Jehovah's Witnesses, and these artificial blood substitutes could be used where donated red blood cells cannot. So, the thing is, why can't we just use haemoglobin? Haemoglobin is the molecule that carries oxygen around the body. It's contained in the red blood cells. It's a protein, so we can purify the protein very well, make it virus-free, bacteria-free, antigen-free, and we can store it for a very long time. The problem is, with haemoglobin outside the red blood cell, is it falls apart very easily. It's very toxic. It's actually very cytotoxic to organs, especially the kidney, also the heart, spleen, the liver, and it causes hypertension. So as soon as you give it, your blood pressure goes sky high. It's not very good for a critically ill patient. Now, some of these you can get past by doing chemical modifications, and indeed, over the last 30 years, there's been lots of clinical trials using these HBOT, hemoglobin-based oxygen carriers, based on chemical modification of hemoglobin purified from either animal blood or out-of-date human blood. And the clinical trials have not been very successful. I'll go through a couple of these. So Northfield Laboratories, for example, they produced the polyheme, which was an HBOT which was from bovine and was chemically modified. And they did 6 clinical trials, and they stopped because of increased myocardial infarctions, heart attacks, pneumonia, multiple organ failure, hemorrhagic shock, respiratory failure. So that's not very good as a blood substitute. We've got Sangar here, polymerized and pegylated hemoglobin, and they went through clinical trials. These were reported to have failed in 2013, and shortly after the company stopped trading. A little bit of a success has been BioPure, the HemoPure from BioPure, and that was used limited in South Africa in 2001 to 2008 because of the issue with getting blood supplies which were HIV-free, but that's no longer used, although it is, essentially the company was bought out by OPK Biotech, which is owned by the Russian government. So there is a license to use it in Russia, but they haven't really used it. But there's interest in it, and again, there's a couple of them going back into clinical trials. But artificial blood has also been in movies. So I don't know if you've ever seen this movie, Daybreakers, Ethan Hawke, William Dafoe. Basically, in this movie, you have a huge population of vampires who outnumber humans, and they keep the humans as slaves for their blood supply to consume their blood. But the humans are running out. The population is getting down, and they're running out of humans. So they try an artificial blood substitute. And they actually have on the film an interview with a Japanese research scientist who was actually developing artificial blood substitutes at the time, in 2009, using artificial cells. So this is exactly what they used in this fictional circumstance. And a bit of a gore alert. The result of this volunteer company's artificial cells was that he exploded. In real life, it wasn't quite that bad, but they failed through, they didn't actually, I don't think they even got to clinical trials, they actually failed during preclinical trials. So there's not been much success on these haemoglobin-based oxygen carriers. And so we did a project quite a few years ago now as part of the EU project with 14 different institutes and universities looking at what the issues surrounding these blood substitutes were, not to create a new one, but to find out the issues. And to understand the issues, you need to understand a little bit about the evolution of haemoglobin. So here's a bacterial cell, very small, it doesn't need a blood supply, it's so small it can just get the oxygen diffusing through the cell wall. It doesn't need a blood supply, and yet it has haemoglobin. And we think the haemoglobin here, the ancestral haemoglobin, is as an enzyme. It's not an oxygen carrier, it's an enzyme doing redox biochemical reactions. Plants have haemoglobin. They have 4 different classes of haemoglobin. You can see some of these in these root nodules, they're red. And these do bind oxygen because they affect the oxygen supply to bacteria in these root nodules for nitrogen fixation. But there are others as well which do redox chemistry, they do enzymatic chemistry. And even in animals, including us, we have haemoglobin and myoglobin, which are the oxygen carriers in the blood, red blood cells in the muscle. But we also have other haemoglobins called neuroglobin and cytoglobin, which we only discovered thanks to the Human Genome Project, because we didn't know they were there until 2001, 2002. And these, we believe, are redox enzymes. So these are enzymatic molecules that do enzymatic reactions, biochemistry. And what's happening with the haemoglobin is that it's going out of control because it's not in its normal environment and these redox reactions are going out of control. So through the funding, what we've done is use genetic engineering to redesign the haemoglobin. So we've added quite a few technologies to make it more stable outside the red blood cell. We've actually used fetal haemoglobin instead of adult haemoglobin. All of you at the moment sitting here probably has about 0.3 to 4% of your haemoglobin is fetal haemoglobin. So you still produce a little bit of fetal haemoglobin, but fetal haemoglobin tends to be a bit more stable than adult haemoglobin as an age group, and we're the first to try and use that. We've also done engineering, which we call these through protein electron transfer pathways, and that essentially short circuits this enzymatic activity, stopping them from doing very much damage. We've also done some mutations, some modifications to the haemoglobin to overcome the hypertensive effects. And this we didn't develop ourselves, this was in collaboration with some people at Rice University in the USA, but these modifications stop it consuming very important cell signaling molecules such as nitric oxide, and this is related to the hypertension. We've also coated the haemoglobin with PEG, polyethylene glycol. Now this has been done before, it's not a new technology, and what it does several things. It helps it become more viscous, much more like normal blood. It stops it from getting through the kidneys, damaging the kidneys, and it stops it reacting with lots of things. And this is FDA approved, this pegylation thing. But the problem with pegylation is that it disrupts the oxygen binding capacity of haemoglobin. I'll come back to that in a moment. But this is just to illustrate what we've done to the haemoglobin molecule. So we have the haemoglobin in the ribbons here, in this protein. So you can see it in the light grey, dark grey. And every single coloured sphere here is a single point mutation which we've done to change the behaviour of haemoglobin when it's outside the red blood cell. I won't go into the details of them, but one of them we've done allows it to be pegylated without changing the oxygen binding capacity of haemoglobin. Because the fascinating thing with haemoglobin is it changes shape. So you can see an illustration of it changing shape here. And if you have a look at the graph of the binding of oxygen to haemoglobin, that's on the y-axis here, the factory saturation. So this has got oxygen bound to 100% of the haemoglobin, this is 0%. It's got this characteristic sigmoidal binding pattern, because with high oxygen tensions, which you have in the lungs, it binds oxygen, and when it gets to the tissue, it releases the oxygen. It's exactly what it's designed to do. But if you pegylate it, add these polyethylene glycol molecules to surround the haemoglobin, you disrupt this change in structure. So when you do that, it becomes more like this green curve here. So it's not releasing oxygen where it should be. And we've generated, sorry about the drawing here, it's a bit more realistic because it's much bigger pegylation molecules than was normally being used, and we only used 2 of them, so we made it much bigger. But we've done it in such a way that it doesn't disrupt the change of shape of the haemoglobin, and it binds oxygen exactly like it should. So we've just been spent last year trying to do the manufacturing process for this HBOC, and when we did it in our laboratory, we have these shape flasks. So we put the gene into bacteria, get the bacteria to grow the haemoglobin for us, and you can see these bacteria here. We spun them down so you can see these bacteria. You can see they're a nice sort of pinky red colour because they're producing haemoglobin. We use a machine which produces about 15,000 psi to disrupt those cells so we can get the haemoglobin out of the bacteria. Then we do various different column chromatographies to purify the haemoglobin. We check for the purity of the haemoglobin. We do some tests, try and modify things, add the polyethylene glycol, get rid of the endotoxins because bacteria produce endotoxins, so we've got to get rid of them before we can put it into patients, and do evaluation tests. What we've done to try and produce more of it is to go from the shake flasks to fermenters. This fermenter is in my laboratory, and it can produce about 40 times the amount of bacteria than the shake flask can per volume. And it's very important for the commercial manufacture of this. We've gone with a company called CPI, Centre for Process Innovation, and they've upscaled it to 100-litre fermenters. And this can usually be upscaled very easily to very massive fermenters, 2,000 to 20,000 litres. And this is a 4,000 litre one person standing right there. At the stage we're here, we have to go through preclinical trials and clinical trials. And for that we need to raise a lot of money from hopefully industrial partners that can do this. And one of the problems of getting us through things like the FDA or the MHRA, which is the equivalent in the UK, is that because these HBOCs have failed a lot before, there's these companies are quite wary. So what we want to do is to prove the technology in more niche markets first, before we go back to the blood substitute. So we are promoting these as oxygen therapeutics. And one of the advantages of these artificial blood substitutes, these HBOCs, is they're very, very small. So here's a hypodermic needle. It's blown up many, many hundreds of times. And you can see all these red blood cells stuck to the end of this hypodermic needle. Here we have a representation of one of these red blood cells, which is about 7 to 8 microns across. An HBOC is tiny in comparison. I probably actually made it too big. It's much more than that. It's about 20 to 30 nanometers, which is 0.00002 millimeters across, very, very small. And this gives it some advantages over red blood cells. For example, if we take a person who has had a stroke or some kind of deep vein thrombosis, blood can't get past that blood clot. But these are small enough to get past the blood clot to keep oxygenating the tissue behind the blood clot whilst the patient gets to the hospital and gets given these clot-busting drugs. There's some evidence, it's still a bit controversial, but there's some evidence that it could be used in cancer therapy as well. Some cancers grow so big that the tumour in the middle becomes hypoxic. And these are quite resistant, most of these are quite resistant to chemotherapy and radiotherapy. Because what chemotherapy and radiotherapy do is they create reactive oxygen species. So if there's no oxygen in the centre of these tumours, it's difficult for these reactive oxygen species to be formed. But these H-box can get into the centre of these hypoxic tumours, deliver oxygen, enabling potentially chemotherapy and radiotherapy to work better. So there's some studies being done on that at the moment. The jury's still out exactly what positive effects that could be. There are already some which are used in organ transplantation. So actually worm blood is used for organ transplantation. And there's a group in France called Haemoglobin that do this. And they, instead of putting tissue on ice, it might be a heart, they did a talk just before the lockdown. They were talking about face transplants. And they, instead of putting them on ice, which has a huge amount of failure in these transplantations, they keep the tissue alive by flowing these artificial blood substitutes in through the tissue to keep the tissue oxygenated. Other things could be sickle cell crisis treatments. People with sickle cell disease, they have a crisis, the red blood cells sickle, they don't get through the capillaries, but can be reversed if these red blood cells are reoxygenated. And these H-box could reoxygenate them. And treatment of shock, so some people who have shock have vascular collapse, so it's very difficult for these red blood cells to get through these narrow blood vessels because these blood vessels are collapsed. But because they're small, they can get through this collapsed vascular system and reoxygenate the tissue. And there's several groups who've been looking at that and have shown a very marked effect on animal studies where they have a vascular collapse. As these H-box systems reoxygenate the tissue, and the animals and potentially the patients will recover. Okay, so just to say about funding, and we've had a lot of funding from the Medical Research Council, but previously we've had from the BBSRC, ICINI, Seacorn Fund, and University of Essex. And there's a lot of people involved on here, so I've got a slide. I don't think everybody is on here, but quite a few people are. So myself leading the research at Essex, but we've had Chris Cooper and Mike Wilson, who are now emeritus. Lots of people at Essex. King's College London are currently working with us. King's College, University College London, Lund University, Palmer University, lots and lots of places. Help with scientific advisors, regulators, and so on, and Centre for Process Innovation for helping us develop the manufacturing process. And thank you for staying with us. Any questions?
Could these increase performance? So could they be used for doping?
Doping, yes, I get asked that a lot.
And do they show up in tests?
Yes, because they're outside the red blood cell. So if you spin down the blood, essentially you have the packed red blood cells and you have the yellowish blood plasma. But if you have these, of course, the blood plasma will be bright red. But no, they haven't been used yet because of the potential toxicities at the moment. And we're doing preclinical studies at the moment to try and prove that these are less toxic than their predecessors. So they haven't been used, but potentially, yes, they could be used. Certainly, what athletes normally do is, of course, go up to high altitudes where they produce more of the 2,3-diphosphoglycerate and more red blood cells. And that helps the oxygen delivery, so they can do it naturally. But yes, it's potentially good. I think we easy to detect. Greg?
You mentioned fetal haemoglobin and
Yeah.
adult haemoglobin. What's the actual difference between them and why have we got two different types?
We've actually got two different types of fetal haemoglobin. We've got gamma 1 and gamma 2. And we're actually using gamma 2. Adult hemoglobin has, hemoglobin has 4 subunits. Adult hemoglobin has 2 alpha, 2 beta subunits. Whereas fetal hemoglobin has 2 alpha, 2 gamma subunits. You even have HbA2, which is alpha 2, delta 2, and embryos have zeta 2, epsilon 2, Gauss